Peter is the chair of therapeutic radiology and the Robert Hunter Professor of Therapeutic Radiology and Professor of Genetics. Peter did his MD and PhD at Yale. I'm sobered to admit that, in fact, we were here at the same time in medical school. Peter has spent his career at Yale, where he has focused, of course, on radiation oncology and has led the group for quite a number of years. And himself is predominantly a laboratory scientist who has worked in a number of areas, but predominantly in the area of DNA damage repair, and is the recipient of an Outstanding Investigator Award that focuses on that very topic. So without further ado, Peter, it's really a pleasure to have you as our first noon speaker in the current era.
Thanks, Eric. I appreciate it. And nice to be here, everyone. Thank you for coming. And it doesn't hurt that there's a free lunch. So that's great. So I'm going to tell you about some of the work that we've been doing for a number of years. And so there's going to be something old and something new related to hypoxia biology and how you might exploit that for cancer therapy. So these are my disclosures. And so many of you will be familiar with the hallmarks of cancer. It's a series of publications from Hanahan and Weinberg and others that have talked about some of the properties of cancer cells. And what I just want to call out here is that tumor hypoxia plays an important role in a number of these, and some of these listed here: altered metabolism, inducing invasion, metastasis. There's a literature on that. Of course, hypoxia causes angiogenesis. But what I'm going to talk about today is our work in which we identified that one of the impacts of hypoxia is genetic instability in cancer. Now, this work started a number of years ago when I was at a Gordon conference on DNA repair mutagenesis, and one of the speakers was Larry Loeb, who's a very prominent pathologist at the University of Washington in the DNA repair field. And he proposed, in the 90s that there would be a mutator phenotype in cancer cells. And this was based on the idea that the frequency of mutations in cancer cells was above and beyond what you would predict from the frequency of mutagenesis in normal cells. So he inferred that there had to be a mutator phenotype. Now, at the time, people were focused on, well, there's going to be mutations in DNA repair mutator genes that is going to lead to the increase in mutation frequency. But as I was sitting in the audience, it occurred to me, well, one of the issues is people do all these studies of DNA repair mutagenesis in cells and culture, but they're growing in a standard tissue culture dish in really nice media at 20% oxygen. And in fact, most cancer cells do not exist in 20% oxygen. They exist in somewhere between 0.1 to 5% oxygen. And in fact, there is a lot of variety in the level of oxygen tension in tumors that has to do with abnormal and fluctuating perfusion, and then as well distance from the vasculature, which creates a gradient of hypoxia. So my proposal was, could this hypoxia that you see in the tumor microenvironment impact genetic instability? Now, one of the first grants that I submitted on this was turned down, and one of the reviewers said, well, that can't be possible because the absence of oxygen will actually reduce the amount of oxidation. And of course, oxygen causes rust and damage to other molecules. And of course, that individual was thinking only chemically, not thinking biologically. So we went on to test this hypothesis, and this is some older data. And the first data set here is a table in which we grew tumor cells that had a specialized mutation reporter, and we grew them either cells in culture or we used them to form tumors in mice. And what we consistently saw was the frequency of mutations was higher when the cells were grown as tumors. Then we went on to study, well, what's the underlying mechanism? First of all, we showed that if you put those cells in hypoxia, you could recapitulate the mutagenesis. So that linked that effect to hypoxia. And we subsequently showed that if you had a damaged plasmid and introduced it into cells that were either normoxic or hypoxic, there was better repair or lesser repair of the damaged plasmid in the hypoxic cells. And this work was carried out predominantly by Jen-Ling Wan, who's now a faculty member at University of Minnesota in radiation oncology. So then we went on to, oh, I'm sorry, I skipped one. So thinking about what we saw in those experiments, we noticed that the pattern of mutations had a fair proportion of changes within repeated sequences, which would be indicative of a mismatch repair deficiency. So we took a candidate approach and looked at DNA mismatch repair gene expression. And what you can see here is that in the mismatch repair pathway, which involves proteins MSH2 and MSH6 recognizing a mismatch, and then MLH1 and PMS2 coming on to mediate of downstream effects and steps in repair. The MLH1 and PMS2 proteins were decreased in expression levels in hypoxia, and that correlated when we put in a specific sequence, repeated sequence that we could probe for mutation. The frequency of mutations was increased in that sequence. So that was the first demonstration at a biological level that there was a repair pathway that was diminished in hypoxic cells. Then we took an unbiased approach and did a gene array analysis of DNA repair expression in hypoxia. And you may see two familiar faces here, Ranjit Bindra, who everyone knows, and Susan Gable, who was then Susan Scanlon, both MD PhD students in the lab and now on our faculty here in radiation oncology. And they did some seminal work in which they looked at the expression of genes that we identified in that screen, including RAD51, BRCA1, and FANCD2 in the Fanconi anemia pathway. So RAD51 and BRCA1 are in the homology-dependent repair pathway. And you can see here on a Western blot that RAD51 is diminished in the hypoxic component, whereas HIF1 as a control is increased. And then here on a Northern blot, which people hardly do anymore, BRCA1 is decreased in hypoxia, whereas VEGF RNA is increased. And interestingly, on a Western blot, you can see the BRCA1 is down, but if you look closely, you'll see the protein is actually shifted a little bit up on the gel mobility, and that has to do with post-translational modification in response to hypoxia. And the same is true with Fanconi anemia. And you see here that the FANCD2 expression is down, and actually there's an increase in the second band, which represents ubiquitination. So what is the mechanism? So it turns out that many of these repair genes have common elements in their promoters that respond to the E2F family of transcription factors. And these factors interact with the pocket proteins that are related to retinoblastoma protein, P130, P107, and PRB. They form heterodimers that regulate these promoter sites. And what we found is that in response to hypoxia, those pocket proteins become hypophosphorylated due to the activity of a phosphatase called PP2A. And this is a common theme throughout this talk that I'll return to several times. And basically the mechanism is that hypoxia causes P130 or the P107 or RB hypophosphorylation, formation of E2F heterodimers, and these are repressive and cause the downregulation of DNA repair. We also found that hypoxia causes changes in expression of microRNAs, and these lead to a cascade of events that regulate DNA repair as well as adaptation to hypoxia on several levels. But I'm not going to go into this further in the talk, just mention it for completeness. In addition to acute and chronic changes, acute and short-term changes in gene expression, Yuhang Liu in the lab has shown that both BRCA1 and MLH1 become silenced with persistent hypoxia due to a specific set of epigenetic changes related to particular histone demethylases. And you can see here as an example, persistent downregulation of BRCA1 and prolonged hypoxia. So just an overview, we found that there are a number of changes in DNA repair pathways related to hypoxia, but predominantly in the homology-dependent repair or homologous recombination pathway and the mismatch repair pathway at the transcriptional, translational, and epigenetic level. In addition, I alluded to the fact that there is acute activation of DNA repair in hypoxia, this is transient, and you can see that in response to acute hypoxia, you get activation of CHEK2, which then leads to a phosphorylation of BRCA1. And actually, as we accumulated data, there's actually a network of signaling events that happens early in hypoxia, followed by downregulation of the DNA repair expression. So initially, DNA damage response pathways are activated and then transcriptionally suppressed and then eventually silenced if the hypoxia continues. So this is basically a summary of a lot of work that shows the scale of time. And in minutes, you get these post-translational modifications, but over time, you get suppression of DNA repair expression in specific pathways at the level of translation, transcription, and eventually epigenetic silencing. Now, people sometimes ask me when I give these talks, why does this happen? Why would the cell do this? And you wouldn't think that a cancer cell that's looking to survive and overcome cancer therapy is interested in downregulating subsets of DNA repair pathways. Well, it turns out that one answer could be related to what people have been doing in the evolutionary biology and microbiology area for a number of years. And this has been pioneered by Susan Rosenberg at Baylor. And what you see here is a study where people isolated E. coli in the wild, a lot of different isolates, and then put them under stress. And the stresses could be starvation in terms of what's in the agar plate as far as nutrients. Could also be certain antibiotics that are not directly genotoxic. And what they found was an upregulation of a hypermutable state in the E. coli that increased their ability to evolve and adapt to the stressful environment. And interestingly, as they started to do the molecular studies, one of the pathways downregulated in E. coli is mismatch repair, and another one is what they called upregulation of a mutagenic double-strand break repair. But what actually happened is they suppressed the normal homologous recombination pathway, and you get an increase in something similar to non-homologous end joining. So very similar to what we saw in cancer cells. So if we want to look at a parallel, the hypoxia-induced genetic instability mirrors what's been seen in E. coli and other microorganisms. And in a way, you could consider cancer as a population of unicellular organisms under stress. Okay, so that's the biology, but now can we exploit it for cancer therapy? So this is work that Denise Heekin in the lab did, where she was the first one to show that hypoxic cells are sensitive to an inhibitor of the DNA repair enzyme poly-ADP ribose polymerase, or PARP. Many of you will know that PARP inhibitors are used in the clinic now for cancers deficient in BRCA1 and BRCA2 because they are synthetically lethal to cells deficient in homology-dependent repair. And of course, I showed you, and I show again here, that hypoxia causes the downregulation of the BRCA pathway, and so those cells are sensitive to PARP inhibitors. So then an MD PhD student in the lab, Alana Kaplan, came and we said, can we exploit that biology for cancer therapy in a pharmacologic way? And our thinking was we would use an angiogenesis inhibitor called cediranib, and it's shown here on the upper right for those of you who are chemically inclined. And it's a small molecule receptor tyrosine kinase inhibitor that was designed to inhibit the VEGF receptor, but actually it inhibits multiple receptor tyrosine kinases not just the VEGF receptor. So we asked, could we use cediranib to cause hypoxia in cancer, in tumors, and then exploit it? So what Alana did was we set up tumors in mice, and we treated the mice with cediranib. Then we isolated cells from the tumors, and this says mouse depletion because it's a human tumor. We deplete the mouse cells, and then we isolate the tumor cells. And you can actually stain them with carbonic anhydrase 9 because it's a marker of hypoxia. And in fact, we saw that there was an increase in the hypoxic fraction in this experimental system from 20 to about 30%. So we took these cells and we looked at expression of the DNA repair genes in the HDR pathway, and we actually saw pretty good downregulation. In fact, we saw downregulation that was too good in the sense that it was more than you would expect from that smaller change in the hypoxic fraction. So it was clear that maybe something else was going on. So what Alana did was we repeated that experiment using that CA9 marker, but instead of just counting those, we actually sorted them and we collected the normoxic fraction and the hypoxic fraction. And what I'm showing you here is the normoxic fraction. And even in the normoxic fraction, homology-dependent repair genes were downregulated, and that was the unexpected result. So what was going on? Well, actually, so we then looked at some of the non-malignant tissues in the mice, and we did not see downregulation. So it was only happening in the tumor. So we looked at bone marrow, liver, lung, and mammary fat pad. And just a little more of a survey, we looked at some other DNA repair factors, and these are in the non-homologous end joining pathway, which we already knew from our hypoxia work that pathway is not affected, and it was also not affected by cediranib. But also in this slide, I'm showing you results of treatment of cells in culture with cediranib, where you cannot generate hypoxia because there's no vasculature or angiogenesis. This is a direct effect of cediranib, and you can see that those pathways are downregulated directly by cediranib. But interestingly, in bone marrow CD34 positive stem cells, we do not see the same downregulation. So that is consistent with what we had seen in vivo. Now, functionally, if we look at cells treated with cediranib and then treat them with radiation and compare them to controls, we see a functional decrease in DNA repair capacity. And you can see here that in normal cells, you will see foci of RAD51 coalescing to repair double-strand breaks. But in cells treated with cediranib, there is substantially fewer foci indicating that that pathway has been suppressed. Similarly, if we do an assay looking for homologous recombination between two fragments of the GFP gene, one of which is cut by an enzyme that can be induced in the cells, we can see that in the presence of cediranib, that recombination event is substantially suppressed, and to a similar extent as what you would see if you knock down BRCA1 or RAD51. But as controls, there's no effect in knocking down the non-homologous end joining pathway proteins Q80 and XLF. Now, we then returned to what Denise had observed in hypoxic cells, and we said, okay, does cediranib sensitize to a PARP inhibitor? And here's the example that shows in the presence of cediranib, there is increased sensitivity to olaparib, a commonly used PARP inhibitor in the clinic. So this is a survival curve on the left, and this is example of molecular markers of cell death and apoptosis on the right. So what these data show is that cediranib treatment phenocopies the BRCA deficiency and creates a synthetic lethality to PARP inhibitors. Now, what's the mechanism here? So I'm not showing you all the data, but one that's particularly striking is we used the papillomavirus E7 protein that Dr. DeMeo will be very familiar with. And the E7 protein was known for a long time to interact with the RB family of proteins and cause their degradation and prevent their interaction with the E2F factors. So when we express E7, we see that there is abrogation of the sensitivity of cediranib-treated cells to olaparib. So this is one piece of evidence that supports that the downregulation is happening by the same method as happens in hypoxia, that is through the RBE2F family of proteins. And so our conclusion of the mechanistically is cediranib has two effects on DNA repair. One is through hypoxia, which was our original hypothesis, and the other one is a direct one through its receptor tyrosine kinase inhibition effects. And in these particular ovarian cancer cells, we linked it to the platelet-derived growth factor receptor. That leads to an upregulation of phosphatase 2A and changes in the phosphorylation of the pocket proteins, and that cascade that leads to suppression of repair capacity. So then we took this into an in vivo model in mice, in which we formed tumors from one of those ovarian cancer cell lines. And this is the tumor growth suppression curve. And the control or olaparib alone are similar. Cediranib has some effect, but now the combined olaparib and cediranib has the most growth suppression, and that's correlated with an increase in survival. Now, when I first presented this at a meeting, I subsequently was contacted by two fairly large pharmaceutical companies. One said, oh, gee, we reproduced that and we're actually working on an even better version of cediranib, and we'd like to come talk to you about that. The other one said, oh, well, we can't reproduce what you did, and actually, we don't like that in vivo model. We prefer a different one. We don't think that one is a good one. So thankfully, Joseph Kim, who I see in the audience here, decided to try a different in vivo model, and that one is called Patients in the Clinic. And in his work, reported in JCO, he did a trial in patients with metastatic prostate cancer combining cediranib and olaparib and saw positive results. So what about other repair pathways? Well, it turns out that, like I said, non-homologous end joining, which you can see here on the right, this is a heat map of gene expression where blue is down and non-blue or red is up. You can see non-homologous end joining is not suppressed, but we do see the suppression of HDR in a different cancer type, in this case, lung cancer. And we also see some synthetic lethality induced to olaparib in this lung cancer line, HCC 827. And then Denise also looked at DNA mismatch repair in a series of cancer cell lines, as shown here in the middle panel, lung cancer, ovarian, and a glioblastoma. And in this case, DNA mismatch repair, as it is in hypoxia, is suppressed in response to cediranib. And this is the heat map, and you can see some Western blots on the right. Okay, so at this point, Gary Altwerger, who is a assistant professor in the GYN oncology department here, joined the lab with an interest in uterine serous cancer. And so we knew that cediranib caused a decrease in DNA repair gene expression in specific pathways, but what impact might it have in uterine cancer? And could there be a role for cediranib in combination with any DNA repair inhibitors or cell cycle regulators in uterine cancer? And part of the reason we were interested in this is that there's a fair proportion of uterine cancers that are deficient in DNA mismatch repair, and the ones that are deficient in MMR have a better natural history and also respond better to immune checkpoint inhibitors. So Gary went ahead and surveyed some uterine cancer lines that we got from Alessandro Santin here in the GYN-onc department. And what he saw was that in the ARC1 on the left and the HEC1B on the right, the mismatch repair receptors were suppressed in response to cediranib at different levels of sensitivity. Interestingly, this ARC4 cell line did not show an effect to cediranib alone. We then tested the functional effect on mismatch repair using a reporter system in which a dinucleotide repeat is inserted at the beginning of the reading frame for the green fluorescent protein. And it's set up so it's out of frame, so there is no green in the cells. But if there's a mismatch repair deficiency leading to a frame shift, you will see green cells. And so what you see here on the right is this reporter was put into those cell lines. They were treated or not with cediranib, and you can see that there's an increase in the GFP positive fraction after cediranib treatment, notably in the ARC1 and the HEC1B, and a tiny one in the ARC4. Now, having seen this functional decrease, we wondered how could we exploit this? Now we knew that olaparib was not known to synergize with mismatch repair deficiency, so we hypothesized that maybe an inhibitor of a cell cycle checkpoint, namely a WEE1 inhibitor, might have some combination effect, because mismatch repair causes replication stress on multiple levels. And WEE1 is a checkpoint protein that regulates cell cycle progression at multiple points in the cell cycle, as illustrated here, intra-S, G2M, and mitotic exit. So we thought there potentially could be an opportunity for a combined effect. So Gary went ahead and did a study to look for synergism. And for those of you not familiar, this is a cell viability assay where different doses of one agent are on one axis and doses of another on the other. And where you see hills that are blue are dose combinations where there's statistical evidence for synergism. So in all three cell lines, we saw that these two agents, one cediranib and a WEE1 inhibitor, which in the slides we mostly call MK1775 or adavosertib, showed good synergism. Now, interestingly, the ARC4 cells had not shown a decrease in mismatch repair gene expression by cediranib alone. But if you can see it here, when we treated them with the combination, we actually saw that there was strong suppression of DNA mismatch repair expression, which you could measure by the increase in cells showing GFP expression using that reporter vector that is visualized here and quantified on the right. The same is true on the HEC1B cells. So what might be going on? Well, it turns out that just like the BRCA and RAD51 promoters, the mismatch repair gene promoters can also respond to E2F and pocket protein transcription factor complexes. And when we looked at retinoblastoma phosphorylation, we could see that the WEE1 inhibitor and cediranib synergized to increase the hypophosphorylation or decrease the phosphorylation of RB, which creates the repressive complex that then acts on the DNA repair gene promoters. So that was one step in the mechanism. But we wondered, well, why is there synergistic killing? So we got a clue from the literature. There is a group now at University of Texas Southwestern led by Guomin Lee, who published this very interesting paper. Excuse me, I'm just get a little water. Where he looked at MLH1 deficiency and its effect on the cGAS-STING pathway. And what he found was when you knock down MLH1, you get an increase in cGAS-STING signaling in the innate immune pathway. And his model was that it's known that MLH1 interacts with a exonuclease called XO1. And this is involved in DNA mismatch repair, but also plays a secondary role in end resection during homologous recombination. When MLH1 is knocked down, XO1 is unleashed and can mediate basically dysregulated exonuclease activity on DNA ends, creating DNA fragments and long single-stranded gaps. And what they showed in this paper is that DNA then gets released into the cytoplasm and can activate the cGAS-STING pathway, which is the sensor of chromosomal DNA and micronuclei and other DNA type fragments in the cytoplasm. So the idea is mismatch repair deficiency, too much XO1 activity, and then activation of cGAS-STING. And one way to score that is the STAT1 transcription factor gets phosphorylated. It's an easy marker of activation of that innate signaling pathway. So we went ahead and looked at that, and we found, in fact, that phospho-STAT1 was strongly induced by the combination of cediranib and the Wee1 inhibitor, as shown here, and quantified its roughly 90-fold increase, which is quite striking. And we were fortunate because Gary is on a K Award with the Women's Health Research Program here, run by Vicki Abrams and Hugh Taylor. And on his mentoring committee was Akiko Iwasaki, who saw this data. And actually, I had the privilege of working with Akiko on a paper which reported that ataxia telangiectasia, ATM mutated mice, which have ATM is known to have cerebellar degeneration as one of its phenotypes, linked the ATM deficiency to hyperactivation of line 1, leading to neurodegeneration. So Akiko said, well, that strong induction of innate immune signaling, that might be related to line 1. So we went ahead and looked into that. Now, what is line 1? It's the long interspersed nuclear elements, which are the most common retro element in the human genome. Most of them are silenced, but some can be expressed. Here's a diagram of the locus, and it encodes 2 open reading frames, ORF1 and ORF2. ORF2 is a reverse transcriptase. And when it becomes activated by transcription followed by reverse transcription, it's linked to genomic instability. And line 1 cDNA in the cytoplasm is known to activate cGAS-STING. And actually, this is sort of a life cycle of the line 1 elements. And the key thing here is, if you can see, you get expression of line 1 RNA, and it gets turned into cDNA in the here in the cytoplasm, and that activates the cGAS-STING pathway. Now, it can also go back into the nucleus and then reinsert and be another cause of genetic instability. So our hypothesis was that there was suppression of MLH1 leading to dysregulated XO1. That could lead to line 1 induction and cDNA production, and then activate cGAS-STING as line 1 cDNA is known to do. So we looked again at the uterine cancer cell lines, and we saw line 1 is activated, increased. So this is a Western blot for line 1, open reading frame 1, ORF1, is increased. And that correlates with our increase in phosphostat1 as seen by Western blot in the combination of cediranib and the Wee1 inhibitor in both ARC4 and the HEC1B cell lines. So this is fitting our model that you get a decreased mismatch of pair leading to line 1 and cGAS-STING and phosphostat1. And so the hypothesis still is that XO1 is playing a role. Okay, now for one part of this, we asked, okay, is the line 1 cDNA causing the phosphostat1 induction? And one way to test that is use a reverse transcriptase inhibitor called 3TC. And this would block the production of the line 1 cDNA from the line 1 mRNA. So if you look here, this is control. This is the combination of cediranib and the Wee1 inhibitor. MLH1 is down, line 1 is induced, and phosphostat1 is induced. Now, if you add 3TC, you see line 1 is suppressed back to baseline, and so is phospho-STAT 1. Now, you can recapitulate that. Instead of doing the combination of cediranib and WEE1 inhibitor, you acutely knock down MLH1 with a siRNA. You get induction of line 1 and phospho-STAT 1. Now, this part was seen by the Guomin Lee lab. What they hadn't realized was that line 1 might be playing a role. And this is blocked, similarly to the case with the drugs, by the reverse transcriptase. So it looked like this is a similar mechanism. Now we went on to test the role for XO1, and in this case we used siRNA knockdown. So you see here, in this case, we're looking at the impact of acute MLH1 knockdown. MLH1 knockdown leads to actually a slight increase in the levels of XO1, but also line 1 induction, phospho-STAT 1 induction, as we saw before. But if you also knock down XO1, you see there's less line 1 induced and less phospho-STAT 1. Then you look at that in the setting of the combination of cediranib and the WEE1 inhibitor. So lipofectamine is just the control for the siRNA to XO1. And you see here that you get the induction of line 1 and STAT 1, phospho-STAT 1, as we saw before with the combination of the drugs. But when you have knocked down XO1, it abrogates those increases. So you see here, there's no induction of line 1. In fact, it's below the baseline, and you suppress the induction of phospho-STAT 1. Okay, so now we return to that synergistic killing, and I want to caution that this is a preliminary result that we are trying to confirm, but it fit the story so well I couldn't help but show it. And what you see here is that there is killing of the uterine cancer cells by the combination of cediranib and WEE1 inhibitor. But when you add the reverse transcriptase to block the induction of line 1, you rescue the cell killing. Now, let's go back to an in vivo model. And so what we did was we tested whether this applied to a tumor model in mice, and we used the ARC4 cells as a xenograft in nude mice. And you can see here, the blue is the control, untreated. These are the two agents independently, cediranib and WEE1 inhibitor. And here is the combination, which essentially completely suppressed tumor growth. And you can see an example at the time of sacrifice of the animals. There's basically scar tissue left in the combination treatment. And importantly, the mouse weights were stable, and so were the blood counts. And this is important because these agents are known to cause myelosuppression. And then we returned to mechanism, and we did immunohistochemistry on those tumors for line 1 open reading frame 1 expression. And you can see that in the combination treatment, there's induction of line 1 expression consistent with the model. Now, I didn't forget about hypoxia, so we're a little bit back to the future, and it turns out Yu Hong, who had done all that work on gene silencing, has been looking at the innate immune signaling pathway, because it turns out that there is some long-term silencing of STING in hypoxic cells. But there's initially induction of phospho-STAT 1. So based on what Gary had found, we looked at line 1 in hypoxia, and it turns out line 1 is induced in hypoxic cells. But that induction can be blocked by the 3TC reverse transcriptase inhibitor. So more to come on that. So just in summary, what I've told you is that hypoxia drives changes in DNA repair and genome instability on multiple levels. That cediranib phenocopies the effect of hypoxia on homology-dependent repair and mismatch repair by engaging a similar signaling mechanism. That creates a synthetic lethality with PARP inhibitors that has been observed in a clinical trial. And we have evidence that WEE1 inhibition will synergize with cediranib to both suppress MLH1 and activate line 1 and innate immune signaling to kill uterine cancer cells, presumably through immunogenic cell death, although that mechanism needs to be fleshed out. And we have recent evidence that hypoxia also engages some of the same pathways, possibly through line 1. So future directions are to dig a little deeper into this mechanism. As I said, I showed some old material, but some new material, so this is all still a work in progress. We want to extend to other uterine cancer models and other cancer types. And we would like to combine with an immune checkpoint inhibitor in an immune competent model. We haven't done that because those were human cancer lines, not syngeneic with the immune competent mouse model. And we would like to test this combination in a clinical trial. So I'll stop there and happy to take questions. Thank you. Tommy. Right. Right. So we haven't looked at that, but that's very interesting. So Tommy was asking about the fact that 3T, I'll go back to the microphone. 3TC is an antiviral. And so what is seen clinically that might be related to what we've seen? And the answer is we don't know, but it's possible that that could be affecting response to therapies. The other interesting thing that Gary has proposed is whether 3TC, if it in fact suppresses innate immune signaling and inflammation in mismatch repair deficiency, could be used as a chemopreventive for people with Lynch syndrome. You know, that remains to be determined, but I think it's something we would like to see if we could model in mice or even in people. Yeah. Right. Yeah, so we haven't looked at small cell lung, and of course, the Schleppen-11 plays a key role in the PARP response. But that would be, you know, very interesting to look at. Yeah. Yeah.
Yeah, you showed a lot of really interesting data about the line expression. This way you showed ORF1 expression.
Yeah.
There's been a lot of study of line 1 expression. It's been very hard to find ORF2, but it's key to your full story, right? So have you found it equally difficult to find any signal that ORF2 is there? Do you know that it's happening because of activity that you see, even though you don't see the protein? I'm just wondering what your experience is.
Yeah, so just to repeat that, the question has to do with, have we, you know, can we detect ORF2 related to line 1? I think as a practical matter, the antibody to ORF1 is a really good antibody, and so that's what almost everyone uses in the literature. And so we haven't looked at ORF2, or if we did, nobody showed me that data, so I don't think it worked. But yeah, that's a good point. Now, we have the indirect evidence that the reverse transcriptase inhibitor suppresses the effect, which makes sense because we're looking for line 1, but we haven't directly measured ORF2. Yeah, Roy.
Peter, I have a question about clinical trials, actually 2 questions. The first, the trial of Joe Kim, the Siderinib, looking at that curve, it looks like there was a positive PFS. So where did that trial go? Has it gone on to a further study? Did any of the molecular correlates confirm some of the scientific advancements we showed?
Yeah, I got to let Joe do it. He's sitting behind you, so he can answer that.
The data was more data for our trials. So I did two trials. One was in prostate cancer, and then the other was in advanced solid tumors, including several tumor types, including small cell lung cancer, non-small cell lung, again, cancer. What you show here is the prostate data. So again, our hypothesis was that we'll be able to see the benefit regardless of your germline mutation setups of the HRG fluctuations. If you told us the price, what we saw was actually we saw most of the benefits in patients with HR deficiency. In other words, the patients with the mutations, for those without the mutation, we're not able to see the benefit of the combination. So actually, our question become actually whether the steroid, whether this actually prevents or delays the peritoneal resistance. So my one question for you is that based on what you have seen so far, whether the steroid, again, it's very hypoxia-inducing agent. We do see this in patients, like it manifests as like the high blood pressure, fatigue, serum-related toxicities. We do believe it's a very strong hypoxia-inducing agent. Whether that actually plays a role in preventing or delaying peritoneal resistance, that's one. And then just to kind of circle back on the small cell lung cancer data, actually we have got image hypoxia with the steroid. We have obtained PET scan before and after the monotherapy image hypoxia induced by the steroid. And we actually saw some early anti-tumor activity in small cell lung cancer. We saw about 30% response rate, and also in non-small cell lung, and also in TNBC as well. In pancreas, we're not able to see any benefits at all. So that's what so my question for you is whether that's going for peritoneal resistance.
Yeah, so that's a very interesting question. And I think what might be going on is that cediranib has a little bit of a broad-based effect on a number of repair factors in the HDR pathway. So some of the tumors that might have, say, a BRCA deficiency, what cediranib also does is suppress RAD51 and some FANCD2 and some other factors in the same pathway. So either one is that HDR deficiency occurs at different levels and the cediranib takes it down a little bit more, or if there's been a reversion, for example, like in BRCA2, there's been some intragenic deletions that cause a reversion in the phenotype. The cediranib can still suppress expression and cause further HDR deficiency. So it could be that that's the susceptible population where cediranib is going to have its most effect.
For your other clinical trial with the pembrolizumab, which looks very interesting, I know you're waiting for more preclinical data, but that's going to be very hard. The animal models are all good, but not so good.
Right.
What are the barriers then to getting an IIT going? Is there a specific disease type that might be best done in, but you do in the phase 1 sector? Where is that going to head?
Well, so I don't know if Gary's in the audience. He's probably in the OR, but he is looking to do a clinical trial in uterine. That's his, that's where this is leading for him. I think for other disease sites, for example, Eric and I were talking about breast, we need to survey based on, genetic characteristics of different tumor types where there's going to be susceptibility, because we know there's variation. So for example, in breast, is it triple negative? Is it going to be, some other subtype? And the same is, we have a hint of a difference in lung between the KRAS driven and the EGFR driven. So there's more to learn as far as what tells us that, these strategies could work. And in fact, Eric and I have briefly talked about, spore projects. And I think something in a spore project could be, trying to figure out what subtype to take this into a trial and then do the trial. Yeah. Yeah. Yeah, so we did do that, and that's in Alana's paper. We did a kind of a I would say medium-sized scale screen. And a fair number do the same thing, especially the ones that strongly inhibit PDGFR. So that's part of what led to our hypothesis of the pathway. So, I can refer you to, there's a table in the supplementary material that has a couple of hundred agents in it. Joe, you had a question.
Add on to that PDGFR question. Just struck, I mean, it's great work and I'm like, wow, but just struck me. Have you looked at cells that overexpress PDGFR and did you get a bigger response or others that when you mentioned the EGFR?
Yeah, we haven't done that enough to say anything. I mean, we thought about doing that and we did some knockout work and some overexpression work. So there is some correlation, but we haven't done a broad survey. And that, for example, could be something that could, go into a project where we, look at, the cell, databases of cancer cell lines and things like that. Right, right.